Thank you. So as Jacob said, I'm Sarah Armour, and I'm going to be talking to you about fatty acid oxidation in pancreatic alpha cells and how it's important for glucagon secretion. So despite discovering glucagon 100 years ago now, we really have no consensus mechanism about how exactly glucose controls its secretion from alpha cells. What we all know is that glucagon is secreted in times of limited glucose. So this is mainly when we're fasting, also during exercise, and we see a hypersecretion in metabolic diseases such as type 2 diabetes. So as you may remember from Jacob's earlier talk, there have been several mechanisms proposed for the intrinsic regulation of glucagon secretion. And the most highly cited of which involves the KATP channels in a similar mechanism to how glucose stimulates insulin secretion from beta cells. And this is depicted in this diagram here, where the increase in glucose uptake and metabolism produces ATP, which closes the KATP channels and causes a depolarization of the membrane. And because of differential expression of membrane channels on alpha and beta cells, instead of stimulating glucagon release, this inactivates sodium channels and prevents the influx of calcium that's needed for glucagon secretion. So some other mechanisms that have been proposed, such as the store-operated calcium channels, which you also heard a bit about earlier when Jacob briefly mentioned them. So here you had the increase in ATP from glucose metabolism actually causes calcium to be sequestered in the endoplasmic reticulum, and therefore lowering cytoplasmic calcium that's needed for glucagon secretion. So you can see that all of these mechanisms that have been proposed are very different, but they all begin with increased ATP production from glucose metabolism. And this is interesting because, A, we know that glucagon is secreted when glucose is limited, and also previous studies have shown us that actually, compared to the beta cells, alpha cells do not readily oxidize glucose. So my project focuses on alternative substrates, namely fatty acid, and how oxidation of this substrate can help to regulate ATP dynamics in the alpha cell and glucagon secretion. So first off, we are looking at ATP dynamics in the alpha cell, and here we use different glucose concentrations and a physiological mix and concentration of non-esterified fatty acids. And we observe changes in ATP through use of the Percival HR sensor, but we express it under the control of the pre-pro-glucagon promoter. So this is only expressed in alpha cells. And you can see that when we add fatty acids to our low glucose stimulation, by the trace here, you see an increase in Percival fluorescence, so an increase in ATP. And you can see that this is significant in the summarized data on the right. Now, to our surprise, and contrary to what people have shown in the past, we actually get a decrease in ATP fluorescence when we increase the concentration of glucose. Now, because we're interested in fatty acids as a substrate for alpha cells, we know that glucose and fatty acids are tightly linked, whereby as availability of one substrate increases, it inhibits the oxidation of the other substrate. So we measured beta oxidation in wild-type isolated islets, and you can see that as we raise the glucose concentration, the beta oxidation is reduced. So to start looking at this more specifically in alpha cells, we used the CPT1A knockout model. So this is knocked out specifically in alpha cells. And again, you heard Jacob mention that model briefly earlier. So just a quick reminder of how glucose and fatty acid oxidation is linked. As glucose becomes more available, you get more pyruvate fed into the TCA cycle through this enzyme here, pyruvate dehydrogenase. And with this increased flux into the TCA cycle, you also get an increased export of acetyl-CoA to be converted through citrate to malonyl-CoA. And malonyl-CoA inhibits CPT1A. So as glucose becomes more available, you inhibit fatty acid oxidation. And you may recognize this graph. which was shown earlier, that shows that fatty acid oxidation in alpha cells is important for glucagon secretion at low glucose conditions. So, using this mouse model, we again measured beta oxidation in islets from these mice. And you can see that at low glucose conditions, beta oxidation is significantly reduced in the islets from the CPT1A knockout mice. And when we look at ATP in islets from the knockout mice and the litamate controls, we see that the controls act as the wild types on the previous slide, with an increase in ATP fluorescence upon addition of fatty acids and a significant drop in ATP when we add more glucose, whereas the CPT1A knockout mice showed a gradual decline in ATP throughout the experiment. And this tells us that in alpha cells, fatty acid oxidation is important for ATP production. So to look at this from a slightly different angle and to confirm our findings, we used a different animal model which had a knock-in of PDK4. So PDK4 is the kinase that's responsible for phosphorylating and inhibiting PDH. So with a higher PDK4 expression, we expect to lower the inhibition of CPT1A through malonyl-CoA. So you would expect a higher rate of fatty acid oxidation in these islets. And what we see when we look at ATP in these islets is that, again, while the controls acted as the CPT1 controls in the wild-type mice, the PDK4 knock-in mice just had a stable ATP fluorescence throughout the experiment. So we didn't see a drop in ATP when we increased glucose levels. We also observed that these islets did not seem to be able to reduce glucagon secretion when challenged with 5 millimolar of glucose. Therefore, it appears that you need this drop in ATP in higher glucose conditions in order to repress glucagon secretion from alpha cells. And we saw this without any changes in glucagon content between the control and the knock-in animals. So we wanted to put all of this together in a mechanism, and our collaborators kindly did some electrophysiology for us. So here you can see two traces. The top one is in the absence of fatty acids, and you may have seen similar traces previously published that show this slight depolarization of membrane potential when you increase the glucose concentration. However, when we look at the same glucose concentrations, but with the addition of fatty acids, while we didn't see any differences in either membrane potential or action potential frequency between 1 millimolar glucose and 1 with the addition of fatty acids, we actually saw that in 5 millimolar glucose, it seems that the membrane hyperpolarizes. And when we add tolbutamide, which is an inhibitor of the KATP channels, we saw a slight depolarization of the membrane, suggesting the potential involvement of the KATP channels in this mechanism. So, in conclusion, we propose a new mechanism of glucose-regulated glucagon secretion, whereby in hypoglycemic conditions, alpha cells rely on oxidation of fatty acids for ATP production. And so when glucose increases, rather than having a direct effect on the alpha cells, it inhibits fatty acid oxidation and therefore lowers ATP production. And with lower ATP production, we would expect that potassium channels, the KATP channels, would open rather than close, and then potassium would flow out and hyperpolarize the membrane. And this would prevent glucagon secretion. So I would just like to thank all of my colleagues in the Knudsen Lab at Copenhagen University and our collaborators in the University of Oxford, from Sweden and Belgium, and of course the Novo Nordisk Foundation for funding the project.
Thank you, Sarah. Questions for Sarah? I think Anna Jonas has a question.
Thank you. Very nice data and clear presentation. I was just curious about your ATP measurements. Did you do measurements in the absence of fatty acids? How is glucose then causing an increase?
Yeah, we did actually do these experiments because we were also surprised when we saw a decrease in ATP. And these data is also on my post, by the way, so you're welcome to come and see that. But what we actually saw was, although we didn't see an increase in ATP, we saw that it remained stable. So we didn't see any change in ATP when we did it in the absence of fatty acids. And I think that there are some studies out there which have shown no change in ATP with addition of glucose, I think from the 1970s, and also some studies looking at fluorescence from FAD and NADPH. So our findings were in line with that. We didn't see any increases, but again, we didn't see this downregulation of ATP.
Do you still think that the reduction of ATP is required for suppression of glucagon secretion?
Yes. Yes, I do. I think because we've also not only done ATP studies, but also secretion studies looking at different pre-incubations. And I think when you have the absence of fatty acids, the pre-incubation becomes very important, because if it's in at a concentration, for example, like 3 millimolar, where fatty acids may be spared, they can then be used in stimulatory conditions, even in the absence of extra fatty acids.
Thank you. Patrick, last question.
I think your data, your data are beautiful. I think it provides a very strong model for the regulation in vivo when there is fatty acids present or throughout. But what about people then, when you culture islets, you can keep islets for weeks in culture and they still respond to glucose. Do alpha cells have large fat stores that can be utilized? Or where do you think the free fatty acids that are beta oxidized, where they come from in your model?
I mean, I think for our fatty acid experiments, we use a very specific concentration, and we have to use, so we bind them to BSA, and we use a fatty acid-free BSA to do this, so we know the exact concentration. I think perhaps if you're in culture, you may get some fatty acids from the BSA that is put into media and such, where they may be able to use that and uptake. I'm not 100% sure whether they have large fatty acid stores, but I think that, for example, if you have a pre-incubation without fatty acids, they could contain the fatty acids in the cell until they're needed. But I don't know how long-term that would be.
All right, thank you, Sarah.